Within the heart of almost every cell in your body lies a microscopic marvel, the nucleus. This tiny chamber houses the molecule responsible for life itself, DNA. Imagine DNA as a long, twisted ladder, the iconic double helix. This ladder holds the instructions for building and maintaining you. Now let's zoom in on a section of this ladder. These sections, called genes, are like individual blueprints within the larger instruction manual. Each gene holds the code for creating a specific protein. Proteins are the workhorses of the cell, carrying out a dazzling array of tasks, from transporting molecules to catalyzing chemical reactions. The sequence of rungs on the DNA ladder determines the type of protein made. These rungs are composed of 4 chemical letters, A, T, C, and G. They pair up in a specific way, A always with T and C always with G. This precise pairing ensures that the genetic information is accurately copied and passed down from one generation of cells to the next. While DNA holds the master blueprint, it remains safeguarded within the nucleus. To build proteins, the cell needs a messenger to carry the genetic instructions from the nucleus to the protein-making machinery. Enter RNA, a molecule similar in structure to DNA but with a crucial difference: it can leave the nucleus. When a gene is activated, a molecular machine called RNA polymerase attaches to the DNA at the beginning of the gene. It then moves along the DNA strand, reading the sequence of letters and creating a messenger RNA, mRNA molecule. Think of RNA polymerase as a meticulous scribe, copying down the genetic instructions from DNA onto mRNA. The mRNA molecule is essentially a transcript of the DNA sequence, carrying the genetic code from the nucleus to the cytoplasm, where protein synthesis occurs. Every 3 letters on the mRNA, called a codon, represent a specific amino acid, the building blocks of proteins. Freshly transcribed mRNA undergoes some crucial processing before it leaves the nucleus. Think of it as quality control, ensuring the message is accurate and ready for translation. During processing, certain sections of the mRNA, called introns, are snipped out, while the remaining sections, called exons, are spliced together. The processed mRNA molecule, now carrying the refined genetic message, exits the nucleus through tiny pores and enters the cytoplasm. It's time for the next phase of protein synthesis translation. In the cytoplasm, the mRNA encounters ribosomes, the protein synthesis factories of the cell. Ribosomes are complex molecular machines that bind to the mRNA and read the codons, those 3-letter sequences that specify which amino acid to add to the growing protein chain. Section 4, Translation: Deciphering the Genetic Code. With the mRNA bound, the ribosome begins its crucial task, translating the genetic code into a chain of amino acids. This process requires the help of transfer RNA, or tRNA, small RNA molecules that act as interpreters. Each tRNA carries a specific amino acid and recognizes a particular codon on the mRNA. As the ribosome moves along the mRNA, it reads each codon and recruits the corresponding tRNA, which delivers its amino acid cargo. The amino acids are then linked together in a specific sequence dictated by the order of codons on the mRNA. Imagine the ribosome as a skilled assembly line worker, linking amino acids in the precise order specified by the mRNA blueprint. This process continues, codon by codon, amino acid by amino acid, until the ribosome reaches a stop codon on the mRNA, signaling the end of the protein chain. The completed chain, a polypeptide, is then released from the ribosome. Section 5, the final touch: protein folding. The newly synthesized polypeptide chain is not yet a functional protein. It resembles a long linear string of beads. To become active, the protein must fold into a unique 3-dimensional shape, a process guided by various forces and interactions between the amino acids. Imagine the polypeptide chain as a long piece of string. Depending on how it folds, it can form a variety of shapes: a helix, a sheet, or a complex tangled knot. The specific shape a protein assumes determines its function. Some proteins act as enzymes, speeding up chemical reactions. Others serve as structural components, providing support and shape to cells and tissues. Still others function as messengers, transmitting signals between cells. The precise folding of a protein is crucial for its activity and its role in the intricate symphony of life.